So this is aerobic, or slow glycolysis. This is picking up from the end of the fast glycolysis, anaerobic. I encourage you to watch that video to get up to speed on where we are picking up today, because today is where the magic happens, where the bulk of ATP is produced, and some really magic stuff happens in the mitochondria. Because today, this is where the magic happens. As mentioned, glucose doesn't do the metabolic work for muscular contraction or cellular movement of any activity inside the body. Glucose helps produce ATP and ADP, and it's the cleaving of those phosphate bonds that contract your muscles. It's what fires your neurons. It's what moves your food through you. So we left off with pyruvate. So the pathology of pyruvate, entry into the mitochondrial membrane. Pyruvate transporter, MPC. Pyruvate's entry into the mitochondria is facilitated by a protein known as the mitochondrial pyruvate carrier, MPC. The MPC is located in the inner mitochondrial membrane and is responsible for transporting pyruvate from the cytoplasm into the mitochondrial matrix. Regulation of the pyruvate transport, the MPC, is regulated by various factors, including the mitochondrial membrane potential and the availability of cofactors like nicotinamide adenine dinucleotide, NAD+. In condition of high energy demand, the increased NADH and NAD+ ratio and decreased mitochondrial membrane potential promote pyruvate transport into the mitochondria. Pathophysiology implications now. The dysregulation of pyruvate transport can have significant pathological implications. For example, genetic mutation or dysfunction of the MPC can lead to a condition known as pyruvate transport deficiency, affecting energy metabolism, leading to various symptoms, including lactate acidosis. Pyruvate is converted into acetyl-CoA through a series of enzymatic reactions in a process known as pyruvate decarbylation. This process takes place in the mitochondria and is a crucial step in the metabolism of glucose, as it links glycolysis, which occurs in the cytoplasm, to the Krebs cycle, citric acid cycle in the mitochondrial matrix. Now there's like two parts of the mitochondria. One's like the inner matrix and the other side's the inner membrane. We'll get to that later. Here's a detailed explanation of how pyruvate is converted into acetyl-CoA. So step one. Pyruvate transport into the mitochondria. We kind of went over this. Pyruvate is a 3-carbon compound from that 6-carbon from glycolysis, produced in glycolysis, needs to enter the mitochondrial matrix for further processing. This transport is facilitated by the MPC, which allows pyruvate to cross the inner mitochondrial membrane. 2. Oxidative decarbylation of pyruvate. Once inside the mitochondrial matrix, pyruvate undergoes oxidative decarboxylation catalyzed by the pyruvate dehydrogenase complex. This complex is composed of multiple enzymes and cofactors. Conversion into the acetyl group. During the oxidation decarboxylation, pyruvate loses a carbon atom in the form of CO2. The remaining 2-carbon fragment is then bound to coenzyme A, CoA, to form acetyl-CoA. The reaction can be summarized as follows. Pyruvate plus NAD plus CoA gives you acetyl-CoA plus NADH plus hydrogen ion plus CO2. The chemical formula for acetyl-CoA is C23H38N7O17 P3S. The enzymatic reactions and coenzymes involved. So pyruvate dehydrogenase complex consists of 3 key enzymes: pyruvate dehydrogenase, E1, dehydrolipoamide, transacetylase, E2, and dehydrolipoamide dehydrogenase, E3. Coenzymes involved in the process include thymine pyrophosphate, TPP, lipoic acid, and flavin adenine dinucleotide, FAD. The conversion of pyruvate to acetyl-CoA is a regulated step in glucose metabolism. The activity of the pyruvate dehydrogenase complex is regulated by various factors, including the level of ATP, NADH, including, of course, being nicotinamide adenine dinucleotide reduced. And the chemical formula for that is C21H27N7O14P2 and acetyl-CoA. High levels of these molecules inhibit the complex, while ADP and calcium ions can activate it, signaling the need for increased energy production. Acetyl-CoA is a central metabolite in cellular respiration. It enters the Krebs cycle, citric acid cycle, where it is further oxidized to produce energy in the form of ATP. This step is critical as it connects glycolysis, which occurs in the cytoplasm, to the Krebs cycle in the mitochondria, allowing for the complex oxidation of glucose and other fuel molecules. In summary, the conversion of pyruvate into acetyl-CoA is a pivotal step in glucose metabolism. It involves the oxidation and decarbonization of pyruvate, leading to the formation of acetyl-CoA, which serves as a key intermediate in the generation of ATP through the Krebs cycle and the electron transport chain. Now let's get into the Krebs cycle. The Krebs cycle, or the citric acid cycle, or the tricarboxylic acid cycle, is a central pathway in cellular respiration that occurs in the mitochondrial matrix. It serves to extract energy from acetyl-CoA, which is derived from the breakdown of glucose and fatty acids. The cycle is a series of chemical reactions that result in the production of energy-rich molecules, including ATP and electron carriers. So, first, the formation of citrate. Acetyl-CoA combines with oxaloacetylate, a 4-carbon compound, to form citrate, a 6-carbon compound, in a reaction catalyzed by citrate synthase. Step 2. Isomerization of isocitrate. Acetylase catalyzes the isomerization of citrate to isocitrate by shifting a hydroxyl group from carbon 3 to carbon 2. Step 3. Oxidative decarbonization of isocitrate. Isocitrate dehydrogenase catalyzes the conversion of isocitrate to alpha ketoglutarate. During this reaction, isocitrate loses a carbon atom as CO2 and transfers electrons to NADH to form transfers electrons to NAD plus to form NADH. Step 4. Oxidative decarbonization of alpha ketoglutarate. Alpha ketoglutarate dehydrogenase complex catalyzes the conversion of alpha ketoglutarate to succinyl-CoA. This reaction also involves decarboxylation and the generation of NADH. Those NADHs are going to be important. Conversion of succinyl-CoA to succinate. Succinyl-CoA synthetase, again, as ACEs are enzymes, if you ever see that ACE at the end, catalyzes the conversion of succinyl-CoA to succinate with a simultaneous generation of GDP, guanosine triphosphate, or ATP. Oxidation of succinate. Succinate dehydrogenase catalyzes the oxidation of succinate to fumarate. This enzyme is unique in that it is embedded in the inner mitochondrial membrane and is also part of the electron transport chain. The electron transport chain is super cool. That's like the lightning in the bottle stuff with spinning molecules and all sorts of crazy stuff. We'll get to that in the end of this video, I hope. Step 7. Hydrate of fumarate. Fumarase, also known as fumarate hydratase, catalyzes the addition of water to fumarate, forming malate. Oxidation of malate. Malate dehydrogenase catalyzes the final step, the oxidation of malate, to regenerate oxaloacetate while generating NADH. Now, the regulation of the Krebs cycle. The Krebs cycle is tightly regulated. Key regulation points include enzymes citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase complex. The regulatory molecules such as ATP, NADH, and succinyl-CoA can inhibit specific enzymes to control the rate of the cycle. In contrast, ADP and calcium ions can activate enzymes to promote the cycle under conditions of high energy demand. The Krebs cycle plays a central role in the metabolism of glucose and other organic compounds, generating energy in the form of ATP and supplying reducing equivalents of NADH and FADH2 for the electron transport chain. This in turn leads to the production of ATP and is crucial for the overall production in eukaryotic cells. But we're not done yet. The electron transport chain, ETC, is a series of protein complexes and molecules embedded in the inner mitochondrial membrane. We talked about how there's the matrix inside and then there's the inner mitochondrial membrane, kind of like around the outside-ish. Still inside the mitochondria, of course. It plays a crucial role in cellular respiration, specifically in oxidative phosphorylation, the process responsible for the majority of ATP production in eukaryotic cells. Let's dive into the details. focusing on complex 1, complex 2, ATP synthase, and ATP produced. So complex 1, NADH dehydrogenase. Complex 1 is first the largest protein complex in the electron transport chain. It accepts high energy electrons from NADH generated during glycolysis and the Krebs cycle, so sucking up those NADHs. As electrons move through complex 1, they provide energy to pump protons, hydrogen ions, from the mitochondrial matrix into the intermembrane space. This creates a proton gradient across the inner mitochondrial membrane. All those hydrogen going on one side like a magnet, right? That's where the charge is. Coenzyme Q, ubiquinon. After passing through complex 1, electrons are transferred to coenzyme Q, also known as ubiquinon, which acts as a mobile electron carrier within the inner mitochondrial membrane, so it's kind of shuffling along on the inside. Complex 2, succinate dehydrogenase. Complex 2 is also involved in the electron transport chain, although its primary role in the Krebs cycle, it oxidizes succinate to fumarate and transfers electrons to coenzyme Q. Electrons from FADH2 generated in the Krebs cycle enter the electron transport chain directly through complex 2, bypassing complex 1. Coenzyme Q, cytochrome C, reductase, complex 3. Coenzyme Q transfers electrons to complex 3, also known as cytochrome BC1 complex. Complex 3 passes electrons from coenzyme Q to the cytochrome C while pumping protons across the inner mitochondrial membrane. So it's shuffling along, right? And each time it spins, it forms all sorts of good stuff. Cytochrome in complex 4, cytochrome C oxidase. Cytochrome C carries electrons from complex 3 to complex 4, cytochrome C oxidase. At complex 4, electrons are transferred to molecular oxygen, O2, which serves as the final electron acceptor, forming water, H2O. You usually respire that water, I think. As electrons move through complex 4, protons are pumped across the membrane. As electrons pass through the electron transport chain, energy released from their movement is used to pump protons from the mitochondrial matrix to the intermembrane space. This pumping creates a proton gradient or electrochemical gradient with higher concentration of protons in the intermembrane space compared to the mitochondrial matrix. So we're forming that charge. Now this is the cool one. ATP synthase, or by ATP synthase complex 5. ATP synthase is an enzyme complex located in the inner mitochondrial membrane. The flow of protons, they want to go back to that, they're attracted to each other, right? They're repulsive of each other, and they're attracted to the negative matrix space on the inside where they came from. So they want to pass through this. Now this is the really cool molecule. The flow of protons back into the mitochondrial matrix through ATP synthase channels drives the rotation of ATP synthase. Like water flowing through a turbine, these electrons are flowing, attracted to the inner matrix. They have to pass through this like water flowing through a turbine, and that coupling of the movement through protons to the synthase to form ATP from ADP and inorganic phosphate. So that spinning of the turbine is taking ADP and forming ATP in this like kind of, assembly line cycle. This process is called chemiosmotic coupling, and ATP synthase acts as a molecular machine that produces ATP as protons flow through it. The exact numbers of ATP molecules produced per molecule of NADH and FADH2 varies due to factors like subtle system and proton gradient strength. Generally, the oxidation of one NADH molecule results in the production of approximately 2.5 to 3 ATP molecules, while the FADH2 molecules yields 1.5 to 2 ATP molecules. In summary, the electron transport chain comprises multiple protein complexes and coenzymes that work together to transfer electrons, pump protons, and create a proton gradient, the lightning in the bottle, across the inner mitochondrial membrane. This gradient powers ATP synthase. That's the molecule that spins when the things flow through it like a water turbine. By ATP synthase producing ATP from ADP and inorganic phosphate. The electron transport chain is fundamental to cellular respiration, generating the majority of ATP used for cellular energy.